We have a workshop for you this morning and this afternoon, nice long session, on the functional genomic, on doing functional genomic screens with the RNAi platform. Before we get started, I wanted to invite you to interject throughout all the sessions that we're doing with questions as we go. We'll try and leave some time at the end of presentations for extra questions, but please do chime in. And it's a long workshop, so I want to keep people energized as we go. You will need a laptop, as I think you were warned, for one of the sections in the afternoon. You won't before that, so we'll hope to have your attention. All right. So first, I just want to describe what we mean by functional genomic screens. Functional genomics is trying to discover gene function in context, in living cells. And we're not going to be able to give you instructions about how to do this in this workshop. This isn't exactly teaching you a skill you'll be able to go home and practice yourselves, because as you'll see, it turns out to be pretty complex to do this, to discover gene function in living cells. But we hopefully will give you a very good taste of what you will see if you go down this road, and prepare you to think about whether you want to do such a project and what it's going to entail. So we have 6 sections for this workshop. An overview section to begin, and then we'll talk about the kind of perturbations one uses for functional genomic screens, how to plan a screen, some of the nuts and bolts details of a screen. Again, we won't try and be comprehensive here. We want to give you examples so that you know what types of issues you'll face. We'll have you go through some of the types of data analysis that you do with functional genomic screens, and then the last section will be how to complete a whole project around functional genomic screening. How do you get all the way to a paper at the end? So I want to start you out with some examples right at the beginning, so you see how these projects look when they work well. So this is the kind of question you could come in with. What's the gene that causes 5q- syndrome? 5q- syndrome is a myelodysplasia. And a postdoc in Todd Golub's lab, now a PI at the Brigham, came in with just that question. He's an oncologist. He knew about 5q- patients with severe anemia. And he also knew that this syndrome had been defined back in 1974 to involve a commonly deleted region on chromosome 5 on 5q, as you might expect. And the commonly deleted region among all patients, they all had different deletions, but the commonly deleted region only included 40 genes. But since 1974, nobody had figured out which was the causal gene, which gene was causing this disease. So Ben Ebert wanted to solve this problem, and he used a functional genomic screen. The key to him being able to do this was that he was able to come up with a cell model of the disease. So he knew that in the disease you saw an increased ratio of megakaryocytes to erythrocytes. And Ben could take hemopoietic cells and differentiate them and assess their relative ratio of differentiation into megakaryocytes and erythrocytes. So the notion in his screen was to take hemopoietic progenitor cells, suppress each of the 40 genes in that candidate genes in that region, and see if he could get the patient phenotype, the increased megakaryocyte to erythrocyte ratio. The big plot here is showing the results he got for each of the 40 genes. Each of the 40 genes along the horizontal axis. Each one of these dots here is an individual shRNA that he used to suppress the gene. And this is the phenotype, the patient mimicking phenotype. And you can see this was a great result for Ben. One of those genes, you suppress it, and you get that phenotype that's seen in patients. The gene was not one that Ben was expecting. It was a ribosomal subunit, not a typical oncogene. Ben was a little bit worried for a while. But he did some things to check this. He, first of all, looked to make sure that he really was suppressing RPS14, his hit gene, and he was. You'll see the theme of how to validate your hits come back again and again. And then he went back and took patient cells that already had that phenotype that he was scoring for in his screen, and he overexpressed RPS14, and he was able to revert their phenotype. He was able to bring the megakaryocyte to erythrocyte ratio back down. So he had it. After those 40 years, a screen answered this question, and pretty decisively. Just one more example. Let's see, come in with the question, which genes in a mammalian cell restrict the ability to infect with flu virus? That's an obviously important question. And a number of groups have actually screened for that type of phenotype, but I'm going to show you one very clear-cut answer that turned out to open up an important area. How do you go about this? Same kind of thing. You need a model, so you have host cells that you're going to try and infect. You infect them with flu that carries a reporter so that you can see if they've been infected with flu. And here are the hits. If you knock down these genes, my pointer's not working, but if you knock down these genes, you're able to, you see much more flu infection. You're getting rid of a restriction factor. One of those hits was an IFIT family member, IFIT3. Turns out this whole family was involved. They're interferon-induced genes. That's what got the screener excited. But it turns out the whole family of genes involved in flu restriction. That's turned out to be a very important direction. So this can work great. You have an important biological question, and you need to investigate to determine causal genes. And you can march through and perturb 40 genes, if that's your candidate list, or the genome, as in the second example that I showed you, and identify which genes will cause an interesting phenotype, as long as you have a good cell-based model and a readout for your disease, for your phenotype, for your cellular phenotype. So, as I said, functional genomics, as we'll define it for this workshop, is to probe gene function in situ, in cells. But it can be very complicated. The two examples I showed you, at that point I'd be ready to sign up for that right away. I can come up with some questions like that, and I want to do some screens like that. But it can get complicated, and that's a lot of what we'll talk about today. Here's some flavors of the complications. First of all, a lot of genes have many different functions. So what you're going to learn is going to depend a lot on the model that you're studying the gene in. Genes can have many different isoforms. The active version of the gene for your particular biology might be a post-translationally modified protein. So that can add complication. And one of the big issues is that the activity the gene will have will really depend on the cell model that you tested in and the environment of that cell. All those details can matter. As we know, a lot of important genes show up in all kinds of tissue types and have many different activities. So here's something to keep in mind throughout the workshop. There are 3 ingredients that are absolutely essential to have a good functional genomics screen. And I've alluded to some of them already. The first is to have a set of reagents that can specifically perturb the genes that you're trying to query. Those could be RNAis to knock down the gene, overexpression constructs, etc. You need a good tissue culture model, or a mouse model. We've done those as well. But you need a good model system in which to probe the activity of the gene. And you need a good readout of what happened to the cells when you perturbed the gene. If any one of those 3 things is not right, is not capturing your biology or doing what you wanted it to, you can get a very wrong answer. So all those elements are critical. That's just about the screening part of a project. The primary screen, here's our 3 elements that are needed for the primary screen. a whole project to use functional genomics actually has more parts to it than that. First of all, you have to choose your projects very carefully. And John will talk a little bit later in the morning about what are good kinds of problems. Then you have to do the part I just told you about, and that's a critical part of the project, obviously. But little appreciated, I think, by some people undertaking this is after you do this primary screen, most of the work usually comes afterwards. You get a list of genes that are candidates to be interesting with respect to your biology, and there's usually a lot of work entailed in figuring out what's going on with those genes, getting all the way to, say, a paper.
Dave, how involved are you in that part, the platform? Is that something that you give the results, and then the collaborator, the initiator of the project goes back and does a follow-up, or are you guys very involved in that part of the process?
So I'll repeat the question put on the microphone. Monica asked, what's the platform involved in doing with people in these screening projects? And I'll answer that in just a few slides. All parts of this is the short answer. So this is just the overview. You'll learn a lot more about the cellular perturbations that one can use from John next. RNA interference and overexpression, I already mentioned, those are genome scale perturbations, and then there are a number of other interesting strategies. There are a lot of different disease models that are tractable. Anything where you can get a good cell-based model, really, you can study. So cancer, osteoporosis, mitochondrial dysfunction, there's a long list. And as I've also alluded to, this isn't necessarily as straightforward as it sounds. The two examples I showed you gave you a clean-cut answer, but it can be challenging to get the right answer out of a functional genomics approach. And we'll get into all the issues around there. The quick overview is getting your model right is absolutely critical, and you'll see a lot of versions of that, examples of that as we go. And the perturbations all have issues around them. They're not perfectly specific to your gene. You have to validate what they did to your cells. So we can't give a protocol for applying functional genomics. It's partly an art. Experience helps a great deal in getting a project to move forward. And that's part of what the RNAi platform does in answer to Monica's question. These are publications that we helped with from 2006 to 2010. So there are a lot of these functional genomics projects where we collaborated with groups to help them finish off these type projects. So in general, what does the RNAi platform do? We do these types of collaborations around gene discovery, gene function discovery projects. We also do a lot of research and development work around functional genomics tools. We try and develop new tools and new applications for existing tools. We have an extensive infrastructure for maintaining a portfolio of reagents that you'll want to use for genomic screens, for RNA interference reagents and overexpression constructs and many other tools, as well as software tools and computational methods. And we have a big inventory of the reagents themselves. There are about 35 people in the platform, and it's a very matrix organization. Everybody's involved in many of these activities, but you can divide it into a few categories. Production of reagents, the libraries, the useful packaging virus to deliver the RNAi to your cells and so forth. Computational and biology and informatics, scientists that interact with screeners, and the R&D effort. And of course, we do all this in collaboration. The platform doesn't focus on a particular biological problem, but we do this all with domain experts in their fields. So I just wanted to introduce you to some of the people who are involved in, the 35 people who are involved in all of this. Some of them you'll see a lot if you engage in a screening collaboration with a platform. Some of them you might not see as much because they're busy making virus down the hall here. But I wanted you to see who some of these folks are that are doing all this work. So I've divided up the group into these same categories. There's a production team that does hundreds of thousands over the years of plasmid DNA preps and virus preps and does library production. There's a screening group that has scientists expert in devising screens and who can help you carry out your screens. I don't have pictures of everybody here, but there are some faces for you to recognize if you tour our platform. So, and this is a subset of the screening group. There's one very big project that we're undertaking where all the screening is happening in the RNAi platform, and we have a big team who are carrying out these screens, so Project Achilles. And this is our software engineering team, and you can infer from the fact that we have a lot of software engineers that managing all the data for our inventory of perturbations and for all the screening data is a pretty extensive task. Then we have the group doing R&D, that's laboratory experiments in R&D. Also computational biology is an important component of our research studies. So if you want information about the RNAi platform, a great place to go first is to our website, our BroadE internal portal. This will give you all types of information about what we do in general and also about how to get specific things from the RNAi platform. But you should come talk with us if you are interested in doing a screening project. If you just want to employ RNAi or overexpression maybe on a smaller scale as follow-up to another to a study that you've done that's not necessarily a screen, just to find out what's available, what could you do. And as I mentioned, we do a lot of research into functional genomic strategies, so some of you might get interested in one of those things. You could certainly approach us to collaborate on that.